event. Thank you, Liz. And so when you ask a question in the Q&A, we will go through and read the question to our presenter because that makes for a better recording. And those of you who are watching this in that way will be able to have the benefit of knowing what questions were asked. If we don't get to your question, or if you have a question about something unrelated to this evening's webinar, please feel free to email us at info@anjac.org. That's info@anjac.org. I guarantee you that email is highly monitored and you will get a faster response by emailing us at info@anjac.org than you will by emailing me directly. And I'm realizing I did not introduce myself. So for those of you who I don't know, my name is Jennifer Coffey, and I have the pleasure of serving as the Executive Director of ANJAC. And I'm joined this evening by a number of our professionals at ANJAC, including Elizabeth Ritter, Sheila Baker-Gurjal, and Randy Rothmel. So our closest friends know all about ANJAC and what our mission is, but for those of you who are newer to us, ANJAC's mission, and we've been doing this for about 56 years now, is really to help municipalities make good decisions about the environment. And we do that primarily by working with environmental commissioners, but also with green team members, open space committee members, elected officials, and we are focusing on helping to support local action, so ordinances, stewardship, planning that supports healthy communities and creates a more equitable New Jersey by looking to protect our natural resources and ensure infrastructure and our lands are functioning at the way that they can best for all of us. We do this and we achieve our mission in a number of ways. We have a resource center that is managed by Sheila Baker-Gurjal, who's with us tonight. We have year-round webinars and roundtables. We have a quarterly magazine that we put out where we share the news that local environmental commissions are implementing on projects and new ordinances, policy, stewardship plans, volunteer actions, because we want to share the word of what's working and have that replicated in as many ways as possibly can for the benefit of all 9 million citizens that we have here. We have two signature programs. The first is our fundamentals course, Fundamentals for Environmental Commissioners. You can find that on our YouTube channel. And so that's all of the what you need to know if you found yourself on an EC and said, oh my gosh, what does this mean and how do I do this volunteer job? So that's all the who, what, why, where, when, and how that you need to know. And we hold that every early spring. So think about around spring training with baseball. And we also have our annual Environmental Congress. This year it will be on September 27th. It will be at Rowan College at Burlington County. So we ask you to hold the date. I believe registration is opened for that. If not, staff will correct me on that. I think registration is open. So you can go ahead and register and ask us any questions that you have about that in the Q&A. We are also your voice in Trenton. So we look out for legislation that will either positively or negatively affect our environmental commissioners, green teams, and municipalities on environmental issues. And we seek to make you aware of it and also work to communicate to the legislature about what we know because of your experience happening at the local level on all things clean water, clean air, clean soils, local farming, pollution, and all those good things. So if you are not on our email list, please do email us at info@anjac.org and we'll make sure to get you connected. This work is supported by people like you. And I know I sound a little bit like public radio or TV when I say that, but it really is true. And it is supported by our membership. So if your environmental commission is a member, thank you. If you don't know if you're a member, you can email us and you can get information about EC memberships as well as individual memberships and corporate memberships by scanning that QR code using the camera app. And so we really do appreciate your support. Anything you can give to keep programming like this happening, free and available to you, we will do our very best to steward your dollars. Fundraising is becoming more difficult. Foundations are unfortunately moving away from environmental giving, And there's certainly a lot of competition in the fundraising space, particularly this year. There's something happening in November that makes it pretty challenging to raise money because there only seems to be so much of it to go around. So we really do appreciate everything that you do for us. We've got other programming coming up to support our priorities. Our priorities are driven by what we hear from you: clean water, flooding, pollution, climate change. These are the issues that we are seeking to find local solutions that you can implement in your municipality. And we work closely with about 300 municipalities. So stay tuned to your email. We've got lots of programs coming up this fall. We've got a series coming up on stormwater management and what needs to be done and what can be done at the local level. Of course, we've got our Environmental Congress coming up on September 27th. So we would love to see you there. We think it's going to be a fabulous event. It is every year, but we've got some very special programming and guest speakers planned this year. So we hope to see you on September 27th. And of course, on webinars through the end of the year as well. So check out the calendar section on our website. And this is how to best keep in touch with us. We're most active on Facebook and Instagram. Those are our social media platforms. And all of our programs do eventually make their way to YouTube when we hold a webinar. If you would like to tell us what you'd like to see in your programs, go ahead and scan this QR code and take our member survey, because we do schedule our programming according to what we hear from you. And with that, I would like to move on and introduce our esteemed speaker this evening, Dr. Wu, who's going to talk to us about HABs, what they are, how we can identify them, and how we can get in touch with our inner scientists. We know that we're only in July, so we're a little more than halfway through summer, and we've already seen quite a few HAB advisories for multiple water bodies. Certainly, there's one that I like to frequent that last week had to prohibit people from going in the water because of a HAB, and so that's unfortunate. It's happening throughout the state. We're seeing it at Weokochik Lake, Lake Opakcong, Carnegie Lake, and 9 others: Farrington Lake, Harry's Brook, Lake Appert, Fox Chase Pond, Spruce Run Reservoir, Willow Pond, Mettler's Pond, Powder Pond, Delaware Lake. And there are probably a few that I missed because the number is increasing. Dr. Wu is going to show us how we can access the information on the latest HAB information that's out there whenever you need it, and how you can help contribute to the data set. Citizen science is really important, but it's also important for us citizen scientists to know what is a HAB and what's not a HAB, because there are some things that look like HABs that are not HABs that we don't need to worry about as much. Dr. Wu is the director of the New Jersey Center for Water Science and Technology and a professor of biology at Montclair State University in the aquatic ecosystems. She's an ecologist whose research focuses on water quality, aquatic ecology, harmful algal blooms, drinking and recreational water safety, seafood consumption safety, pollution transport control, microbial source tracking, and ecosystem restoration. So we really appreciate her bringing her expertise and knowledge to us tonight. In addition to researching and conducting research, Dr. Wu directs a water analysis lab that is DEP certified, organizes a citizen science program for HAB monitoring, and coordinates environmental education and outreach programs. She's the author of more than 50 articles and books, educational DVDs, and also holds 3 U.S. patents on innovative technologies for water treatment. I don't know how we don't know each other, but I'm really thrilled to have you here tonight. So I hope our paths cross very frequently moving forward, because all the stuff you're doing is certainly of interest to me as somebody who serves on the State Water Supply Advisory Council, and as someone who wants to get more citizens involved in science. Certainly our EC members are ready to be engaged, and we appreciate you telling them tonight how they can best go about doing that. So with that, I will turn it over to you to share your screen and walk through tonight. So Sheila, did I miss anything? Do I need to add anything?
No.
Okay. So if you use the Q&A function to ask questions tonight, that's going to be best. And I see some people have used the chat, but the Q&A does really enable us to keep track of your questions best. So with that, I'm going to turn it over to you and say thank you again for being here with us.
Thank you, Jennifer. And thank you for the opportunity to invite me here I'm so happy to be here to share about harmful algal bloom in New Jersey. I just want to make sure that you can all hear me well. If somebody can give me a thumbs up, because I
Yep,
know.
we can hear you and see your slides.
Thank you. I'm sorry. I apologize. My puppy is barking behind me, so sorry about the interruption. Um, as you can see on the picture, that there are more places around the state across the globe that have been turning green due to the hot summer weather, and that is a result of harmful algal bloom. As you can see on my title, I put it down as cyanobacteria harmful algal bloom because the problematic issue here is actually cyanobacteria. But algae is really a common term when we refer to a very diverse group of microorganisms. And here we are really going to zoom in. It's about cyanobacteria. Cyanobacteria is actually a very natural part of ecosystem. It's very widely distributed across both aquatic and land. They can actually survive on land as well. They could be like coated along the wall, or they could actually be on the rock, in the soil. Not only can be found in water. They are actually very important part of the aquatic ecosystem because they are food sources for other organisms. So they play a really critical role in the aquatic ecosystem. The other reason they are very super important is because they produce oxygen through photosynthesis. They are part of the photosynthetic organism group. And they are very numerous in the ecosystem. So they produce a lot of oxygen and which make it critical for the ecosystem. Well, some cyanobacteria, not all, but some cyanobacteria also have the ability to fix nitrogen. So it's a good and a bad. Having the ability to fix nitrogen really will help with ecosystem that doesn't have enough nitrogen or enough nutrient and is not as productive. However, it could be problematic in ecosystem that has plenty of nutrient around, and it does add that competitive advantage for cyanobacteria to outcompete others when nitrogen is a limiting factor. Cyanobacteria comes in all shapes, sizes, and colors. So I have some example here to show you how diverse morphologically they could be. They could be single cell. They could be single cell that living in kind of like a water sac, such as microcystis that is at the bottom right corner of the picture. So each of the dot inside that look like kind of like a water bag is one cell. So lots of cell clustered together in there in this random pattern. Could be one, we call it colonial, and they just kind of get together. But they could also be a single cell without that water bag type of structure, and they could be like scattered around in a single cell format. They could form like a cluster like this Unocinia at the central bottom picture, and they kind of cluster like a ball together. They could form like a chain together, or they can stack together in filament, like the other picture on the left that you can see. They could also vary in color. A lot of people know cyanobacteria as blue-green bacteria or the old name blue-green algae, but they are actually bacteria. And the reason they are called blue-green is they tend to be in blue-green color. However, they could also be in pink. They can also be in more like reddish or neon green. So it does come with all shape, size, and colors. When they grow and accumulate into too much biomass, they could become harmful or not favorable environmental condition. And some of the reason for that to happen, of course, it could be when there are too much nutrient available or when we have hot temperature. And again, that tend to be the situation happen during the summer or like the recent days that we have heavy rainfall, and the runoff can carry nutrient into a water body, and we have really hot temperature. So that could be a recipe for harmful algal bloom. Well, the other reason could be hydrological stability. So for example, when lake is stratified, or when we are going through a drought and the river water level is low, or when the river water level is flowing very slowly, or when a lake has a much lower fresh through situation, and that can also, increase the reproduction of cyanobacteria. Well, climate change, of course, is not really helping because climate change does come with warmer temperature and periods of drought, slow flow, nutrient loading from intensified rain event or runoff. So those actually are not cool because those tend to be the factor that will either cause harmful algal bloom or intensify the magnitude of harmful algal bloom. So they are not cool. We don't like them because cyanobacteria not only is an eyesore, but it also can produce cyanotoxin and other irritants. And that can cause serious health effects in both people and animal. We can group those toxins into different type based on where it targets. For example, some cyanotoxin can target liver, some of them will target nervous system, and some of them would target like skin or mucous membrane, or some of them just kind of make you to have allergy reaction, rashes, stuff like that. So some more serious impact, of course, can come to more of a long-term exposure or more acute cause that could happen. Um, According US EPA, it could actually cause people to have liver failure, or it could cause people to have kidney issue, or in more severe situation, it can cause human death. And then some people have been linking cyanotoxin to some of the neurological symptoms such as Alzheimer and Parkinson. And not to mention a lot of time, the symptoms are so close to common allergy, flu, or stomach flu. So it could really mix up with all other possibility. So a lot of time people were exposed to cyanotoxin, did not realize the symptoms were actually caused by cyanotoxin. It's very easy for them to think about, I might just have a flu, or I might just have a food poisoning, or I might just have an allergy. And that's one of the reasons how it has been going under the radar for a while until we see more and more cases and more study have linked it to the cause. And of course, it's not just harm human, it's also harming the environment. Because when you have too much harmful algal bloom or cyanobacteria growing, just like the one that's showing on the picture, it could also create like a blanket situation for the water body. And that can interfere with oxygen diffusion into the water. And at night, when you have a high amount of biomass, does respiration at nighttime without photosynthesis because of lack of sunlight, it could consume oxygen. So the dissolved oxygen level can drop to really low at nighttime. And it is pretty common for us to see that during a harmful algal bloom event, the dissolved oxygen level is really high in the afternoon after a photosynthetic period of time, and then drop low at night after respiration, rapidly using it up. And as we all know, dissolved oxygen and pH also could be linked together. And photosynthetic activity and respiration also either use or consume carbon dioxide. So just like dissolved oxygen level can go through up and down throughout the day within the 24-hour cycle, we can also see the pH level going through the up and down cycle within a 24-hour cycle. So those extreme high and low dissolved oxygen level, extreme high and low pH level are really not suitable for aquatic biota. So it could really be harmful for other aquatic biota or plant material. Well, the toxin typically are hold within the cyanobacteria cell, with some that could be also on outside the cyanobacteria cell and attached to the cell membrane. But most of them were actually inside the cyanobacteria cell. So you could have low amount of free cyanotoxin. However, naturally, when the bloom decay and the cell broke open, or when we wanted to treat a water body when there are too much algae growing and we start like a chemical treatment, then you can break the cell open. Or when human or animal ingest water and ingest cyanobacteria, then naturally through our digestive system, it will break the cell open. So when cell break open and toxin got released, and all those scenarios will result into toxin become available and start to have harmful impact on organism. So how do human and animal be exposed? Well, when we are in the water body, we could naturally be in contact with the HAB infected water. But cyanobacteria and cyanotoxin can also be airborne. For example, they can be dissolved in a water vapor. So when you are near the water body, there's also the possibility you might be breathing in cyanotoxin that's or cyanobacteria that's in the water vapor. You can also accidentally swallow water that infected by harmful algal bloom while you are swimming or when you are actually, drinking water or eating food that were coated or contain harmful algal bloom. And of course, you could have direct contact when you are doing water recreation. And animals that are living in the water also can be exposed to. Well, unfortunately, we consider children are at much higher risk because they have smaller body weight, but they have much higher water content. And particularly like bottle feeding children, they are drinking a lot more liquid. So if the water is contaminated with cyanotoxin, there's much higher chances for children to get sick. Well, when it comes to recreation, when children are using water for recreation, they have a much greater tendency of swallowing water, as well as they had this hand-to-mouth behavior, putting their hand into their mouth. And by doing so, they could actually be drinking water, well, swallowing water, I should say, or by licking their hand and have the toxin or bacteria ingested into their body. Well, animals that live in water body also could be at great risk because they could be drinking, swimming, or feeding in the water. And not only wildlife, but also pets after they are swimming in the water. They could also have their hair or fur or feather coated with cyanobacteria. And then while they're grooming themselves or licking their fur, then they could be ingesting those cyanobacteria as well. So if you have a dog, then you like to take your dog to a pond that you got to really watch out because a lot of time we let dog go swimming in the water and they could be covering themselves with cyanobacteria and then coming back they could be licking the material of their fur. So that's definitely one thing to keep in mind. Well, a lot of people say they have never seen HAB issue in the past, and why did they show up all of a sudden? Which is not true. They are definitely not a new phenomenon. It has been going on for a long, long time. As a matter of fact, our neighboring state, New York, has started the monitoring program back in the 80s. They were harmful algal bloom situation all around the globe, all the way back to the 70s and 80s. So because of that, we really want to say it's not new. We just did not notice that. So for example, this is the picture of Branch Brook Park in Newark back in 2011. And of course, that top layer is cyanobacteria. This is Middlesex Reservoir in 2015. And again, that's a picture covering cyanobacteria. This is Orange Reservoir in 2017, another picture, a harmful algal bloom. And those are just some examples showing you that it's not a new phenomena. It is only till about 2019, 2018, 2019, when we started to see that, ooh, there are signs putting young by NJDEP, and there are newspaper articles telling people that stay away from those water body, and such as this one talking about, stay away from Greenwood Lake. This is not a new phenomenon. It is only that we are starting to pay attention to the problem. In 2019, we started to put sign out there telling people that don't swim, suggesting you to not use the water body for recreation. And that is when we notice it, we start to see more and more pop up. But it is definitely not a new episode. It has been going on for a long time. It's not only affecting recreation water, it is also affecting drinking water, source water. So a lot of our drinking water, source water also got affected by harmful algal bloom. And this is an example of Manasquan. Manasquan has several episodes and it has been under treatment for many, many years. So we do know that it is definitely a water body that on our agenda. So Jennifer just mentioned Spruce Run. That's another reservoir that we know has repeated year after year and even over winter bloom that we have observed. And those are just two examples. And there are many other drinking water, source water that also are with repeated harmful algal bloom episode that happen year after year. Not only reservoir water, but we start to see that they are well water, private well, shallow well, most of the time, that are linked to some of the water body nearby. So again, if you do have a water body nearby that are having harmful algal bloom ongoing, if you have a shallow well that hydrologically is connecting to the water body, you could actually possibly that draw the water from the water body into your shallow well, and you could have harmful algal bloom also in your drinking water, well water as well. So in New Jersey, Department of Environmental Protection is the primary agency that are giving us any sign or alert and to tell us what to do, or I should say recommendation. So NJDP actually have this tier system, and then based on different condition, and there are criteria associated with each of the condition, and you will place the water body under different alert. And as you can see that on the screen, I kind of highlighted with the red box. When the cells level is lower, then you do not have to worry. But once the cell density is going over the threshold, which could be 80,000, depends on whether they are toxin present or absent. But basically, you could say when you have below 80,000 cell per mil of cyanobacteria, and you did not find toxin going over, then you're okay. But once you have either any of the toxin going over the threshold, and there are 4 toxins listed by the state of New Jersey, so any one of the 4 going over that threshold, or when you have a cell count that's above 80,000, then we know not a good situation. We really have to watch out, because that's what advisory is here, telling you that we really need to watch out because it could be potentially dangerous to human health. We should close the beach. We should stop using the water for recreation. But just keep in mind that NJDP is not the regulatory agency. It is there to provide you with recommendation. That's why the word recommendation is listed on the table. They can help you let you know what kind of alert level your water body is. They can make recommendation toward what should be done, but the decision really stay with whoever is the owner of the water body. So if you have a private lake, whoever is governing that private lake will have to take on the responsibility. If you have a swimming pool or you have a beach, maybe your county health department or your municipality health agency will then have to be the one making such a reinforcement. The enforcement power is on the owner or the government party of that beach or recreation water body. So when the toxin level go over a certain threshold, then the water is really very dangerous, and definitely we should close the beach and not allow for recreation of any kind. So whenever there is an alert level recommended, there will be a sign associated with the alert level, and that we'll talk about it in just a minute. But just so you know that based on the criteria, again, it's either based on cyanobacteria cell count or based on the toxin level. So there are different methodology that people use to actually figure out what is the cyanobacteria level. It could be very simple and doesn't require a lot of training or tool, or it could be a lot more complex and needs specific training and equipment. Some of them you could do, and that could be a very nice first line of monitoring that citizen scientists or any people, any stakeholder, any concerned citizen can do. For example, visual assessment. So those are some example that you could see those signs, such as if you see dot, you see some of the foam or strike or scum or on the water body, or simply when your water is turning green. So visually, you can notice there are ongoing harmful algal bloom, and that would be the first sign, and anybody can do. There's also another very simple way that we call jar test. And jar test is really simple and anybody can do with a mason jar or any kind of transparent jar that you can use. It is for planktonic cyanobacteria, so any water body, not benthic, not growing at the bottom, but free floating in the water body. Only very simple 3 picture as you can see here. You take a jar, you collect your water, shake it, make sure that it's well mixed, but you also don't want to shake it too much to break the bacterial cell. So you basically mix the water, and we usually say by reverse mixing it. You mix them and allow it to stand there resting for 15 to 30 minutes. And after that, you can observe where that green color is located. If the green color tend to be at the bottom, such as the picture on the B picture, then mostly those are not cyanobacteria, because cyanobacteria tend to be a little bit smaller and lighter, and they like to flow on the top of water, such as the picture C. So if you see that your green mass settle down at the bottom, most likely you're okay. But if you see them either suspending in the water or floating on the top, kind of like the picture on the sea, either have a layer on the top or well mixed, then you might have cyanobacteria harmful algal bloom. So very simple test. Well, a lot of time people are having issue between planktonic cyanobacteria or filamentous green algae, which is another very common algae in most people's water body. Filamentous green algae are not toxin-producing algae, so they are not really hurting human health. They could be an eyesore. They could make your water body look nasty and you don't want to go swimming, but it's not really necessarily going to affect human health or animal health. So it is a good idea to be able to tell that whether you have a cyanobacteria or a filamentous green algae. Very simple test because they are filamentous green algae. That means they stick together and they link together and they look more almost like a hair or film. So you could simply use a stick. That's why we call it a stick test. And you try to poke or try to kind of go through that green mass that you have seen. If it's cyanobacteria, most likely it would just kind of disappear and without sticking on your stick. But if it's a filamentous green algae, you will have either a film or hair-like or the whole entire mass that you can pick up with your stick. So with that, you can tell cyanobacteria with green algae apart. So you'll be able to tell them apart. And again, a very simple stick test that anybody can do. The other way to test it, and it's a good numerical way, if you don't really trust that your own observation, then we can trust the meter. So a simple phycocyanin meter like this one that you're seeing on the photo could really be a wonderful tool. It's not very costly. It's about $1,500 for 1 meter, and it requires very low level of calibration that you could absolutely do it. NJTEP also have a loan program that you could actually loan the meter at the beginning of the season, and they will be able to provide that to you. Our office also have a loan program. So if you are not able to get it from NJDEP or you rather come with us, we'll be happy to work with you as well. So basically, it's a meter that measure the photosynthetic level of phycocyanin, which is a pigment that cyanobacteria use to do photosynthesis. And freshwater cyanobacteria are more dominant with phycocyanin, not chlorophyll, like we always heard about chlorophyll. So instead of chlorophyll on plants, it's really phycocyanin for freshwater cyanobacteria. But saltwater cyanobacteria have a different pigment. So this machine specifically measuring phycocyanin is only good for freshwater. And if you have a saltwater system, there are phycoruthrin meter that you could use for saltwater system. So it's a handheld meter and you dip it into your water, just like the one that you're seeing on the picture. It requires very simple calibration, which anybody can do with your tap water before you take the reading. And the meter can read anywhere between 0 to 99. If you have it less than 13, NJDP will say that, ooh, you're good. If you go over 13, then you really have to kind of watch out and start to be alert. And I usually tell people that, if you have single digit, you're okay. But if you go to the over single digit, you start seeing double digit, then you really have to watch out for that. And again, this is something that you can either get a loan or you can purchase one if you have budget available for your own municipality. And it's a very good tool that you can use anytime you want at multiple locations in the same water body, multiple times throughout the day. Well, of course, another way for us to really go into, let's say, regulatory, right? We want to apply for the alert level, want to shut down your recreation activity. We have to go with a more scientific method that we can really quality control and back us up. The NJDEP use microscopic method, and we also recommend using microscope to count cyanobacteria cell and ID cyanobacteria under a microscope, as they are so small. They could be in the micron range. And so it's necessary to look at under the scope to properly identify them morphologically. And once you identify morphologically, you can also count the cell. If you remember how the cyanobacteria could actually be in a filament, you actually have to count each of the cell in the filament, or they could be clustered together, and you actually have to count each of the cell in the cluster. So it's not an easy process, and it is definitely required trained personnel to do so. But this is the way that we currently use in order to set up the alert level. Another criteria that people use is cyanotoxin, and there are currently 4 different cyanotoxin that have a threshold attached with it. And it could be simple, minimal training required, or it could be very complex and require specific equipment. Strip test is something that anybody can do. If you want to have a visual of the strip test, it's kind of like a pH meter or a pregnancy test. So you put the strip test paper into the water and you observe the color changes. And the more pink it is, the more safe it is. So it is, again, observe the color changes and using different shade of pink to decide that what the toxin level should be or could be. Then they are biochemical assay that we call ELISA, which is currently what NGDP use and what EPA recommend us to use when it comes to deciding or assess the safety level for people to use a water body for recreation. So this method is a biochemical method we call ELISA, and it's a biological assay that uses reactive protein or antibodies to detect and quantify cyanotoxins. Currently, microsystem, which is one of the four that has a threshold attached to that, does have a EPA method that is currently available, and it's considered a certified analysis. So you could actually find a laboratory that's certified by EPA or DEP to do such a testing, so you will know that the result could be trusted. So you could actually use that to make your informed decision whether a water body is safe for recreation. And this is definitely a recommended method to quantify cyanotoxin. So when you have a situation that either through visual assessment or through your jar stick test or using your phytokinetics meter reading, and you suspect you do have a harmful algal bloom, the next step that you could do is to report that to NJDP. So NJDP currently does have this whole entire page to report a ongoing harmful algal bloom incident. And once you file this report, they will then send somebody, one of their staff member, to the water body and to actually do the data collection, sample collection. After that, they will then put the results on their website, and it will, of course, associated with a HAB alert status, letting you know whether you do or you don't have a confirmed HAB. And with that, they will also put up this sign, and based on different alert level, they are different color of sign. So orange is an advisory, and then of course you have warning sign, or they have danger sign in different color, red or black. And on the side will also be telling you that can you swim, can you do secondary recreation that doesn't put your head in the water, can you wade into the water, can you allow your pet to go into the water, or can you drink or eat the fish? So all those are super important, and it just gave you enough information to tell people if you could use the water body or you should stay away from the water body. I included some resources here, and if you want to read more about harmful algal bloom or get to know more about how to prevent or manage harmful algal bloom, there are some links here that you could visit. Number one, of course, there is the NJDP HAB program website that will have all the detailed information about the methodology, about the health impact, about alert levels such as those that we have gone through today. It will also give you a list of locations where are ongoing and where are not confirmed that you're safe to do. There's also a HAB lake management guidance, which is a documentation along with presentation to let you know that how can you find out the status of lake. For example, how do you monitor a lake? What are some of the methodology that you could do to find out how healthy your lake is? And what could you do to reduce your nutrient level or to actually treat your harmful algal bloom if you do have an ongoing harmful algal bloom episode? The Interstate Technology Regulatory Council is this multi-agency and whole US initiative to really provide people with the most up-to-date, most advanced hot science. And it has a lot of information all the way from the ecology, how to identify, how to monitor, how to treat, and how to do the management, the regulation, stuff like that. So it is a really good resource if you want to learn more about HOP. EPA, of course, also have HUB website and our own center, New Jersey Center for Water Science and Technology. We do have a New Jersey phytoplankton lab, and that also have information up there available for you to read about harmful algal bloom. And all the links are here. And I also provided a link, so hopefully that it will be an easy access for you to go visit those websites. So NJQuest does have a citizen science program. That's one of the information I would love to share with you all. And what we do is we work with community group, or we work with anybody who's interested in working together, such as neighbors, to monitor the harmful algal bloom condition in the water body of your choice. The goal is you will be able to, as a citizen scientist, to alert your community member when there is a hot present, to alert them and tell them stay away from water, or let them know that the water is actually safe for you to go and use it. We really envision that all our citizen scientists will be able to share the knowledge about harmful algal bloom, about its potential health risk, how to protect them yourself and your pets from harmful algal bloom impact. We love and we trust our citizen scientists will be able to share that knowledge to their community, their friends and family. And that's really our ultimate goal, is to be able to spread the knowledge to as many people as possible. So in 2023, we had 33 citizen scientists from 5 different teams that monitored 20 sites at 11 water bodies. And we trained them to monitor water pH, temperature, dissolved oxygen, specific conductance, and they also take the Saki depth reading. We teach them how to identify cyanobacteria, how to count cyanobacterial cell, so they could actually have a better understanding to what cyanobacteria look at. And we also provide them with one of those meters so they can take the reading of phycocyanin. So they started working in June, and they do it all the way till November. And we do have 2 guides. It's visual guide, both have online version as well as hard copy. So there are links that's also provided, and you're definitely welcome to visit our website. And it doesn't have every single taxa, but it has the most commonly found cyanobacteria in New Jersey, as well as other phytoplankton and aquatic biota that you find in Jersey. The goal is for you to look under the scope. You can distinguish between cyanobacteria and other non-cyanobacterial taxa, such as green algae or diatom. And then with the cyanobacteria, you will then be able to learn who they are under the scope, and then be able to do the cell count to find out what is the hot risk level that you are at. If you visit our website and you're interested in getting a hard copy, you could also submit a request form, and we'll be happy to send you the ID guide. So what did our citizen science find out in 2023? They did a cell count, and again, we provide them with microscope. We provide them with a counting chamber that they can look under the microscope. And you do see that that red dotted line, that's the 80,000 cell density threshold that NJDP established. So in some occasion, and which is more frequently than we want to see, we do see that cyanobacteria are actually above that 80,000 threshold line. So again, we of course was hoping that everybody have safe water for recreation, but unfortunately, that's not always the case. So they will be able to, those citizen scientists that are working with us, were able to inform their community member that, hey, it's not a good idea to jump into water. Our water is not really safe for recreation, which is exactly the goal of our citizen science program. And from what they have done, our citizen scientists found out that there are some cyanobacteria that are more frequently and commonly observed in New Jersey, which is kind of the same as what we have seen in in all the water body across New Jersey. Dolichospermine, Aphanezomenon, Aphanecapsa, Unokenia, Microcystis, and Atheke are some of them that have been commonly seen. And I want to point it out here because those are microscopic pictures that my staff have taken under the scope. So it's very clear. But you could still see that morphologically, they are very diverse and very different. And so it's really just a fun journey for anybody who's interested in observing cyanobacteria or phytoplankton. It's really a lot of fun. So I hope more and more people are interested in doing so. So in 2023, we also rolled out a free cyanotoxin testing program. And we had 6 community groups that partnered with us. And what they did is they collect water sample and they send it to Montclair and bi-weekly from July to October. And so every Monday, they send us water sample. Then we use the certified method, and we are actually a certified laboratory. So we want to say that you can completely trust our results. So we tested 14 sites at 9 water body. And again, those were collected by the community member. And we simply just let them know what the results are. And this was a free program that we rolled out back in 2023. So what we did is we created this HAB data dashboard, and it's a GIS-based data dashboard. So anybody can actually visit this site, and it's actually free access to anybody. So you can visit this data dashboard, and you can click on the water body of interest, like those dots over there, and each dot is actually done by one group, that one community group that work with us. So it's color coded that way. Then when you click on that, it will pop up on all the data entry from all the data point, all the sampling event, and then what are the results, what's the toxin level, what's the DO, and what's the pH, and what's the Secchi count, for example. So that we consider is the easiest way for people to access data. So any other community member that, you live by this lake or you want to go and use a lake for a daily recreation activity, you could pop on the website and learn the condition, and then you will be able to make an informed decision on whether you want to do your recreation work in that water body on that given day. So in 2024, we continue our citizen science program. And this year we have 5 groups that work with us. And this is an ongoing group, and they just started their data collection, as you can see. And we anticipate will continue until October as well. So in addition to the citizen science program, we are definitely a public institution, and we are here to also work with a community member. We have been having an ongoing outreach event with a lot of different local organizations. Our goal is to be able to provide scientific and technical support to community partner, any the organization, grassroots organization, municipality, and we focus on water quality, pollution reduction, flood mitigation, anything that's water related, recreation, drinking, wastewater, and all those are our goal. And again, we are here to provide technical support. We do have, as mentioned, we have a certified water quality testing lab, and we can help you test your water if you have a certain testing need as well. We do have an ongoing education program as well. We do all different kind of community event or classroom visit. So we routinely go out to classroom or any of the science fair, any of the Earth Day event, things like that. And we do both on site or in classroom or field trip with different age group. And again, it's a topic centered around water science and water conservation. So if any of the school district or any of the community that does have an event coming up, please feel free to reach out to us. And again, we can do classroom, we could do table, and we can do field trips. So we definitely would love to work with you on that. We also do teacher training, and we have host many, many workshop, and again, center around water science, water conservation, and harmful algal bloom. Those are usually what our teacher workshop is about. And as you can see that we don't just give lecture, our goal is to be able to provide some activity that teacher can actually take those activity and actually use it in their classroom so it can have a deeper impact through hands-on experiential learning to boost students' interest and to engage students in water science and conservation. And all those education and teacher training events are free of charge. So if any, again, municipalities, school district are interested in so, please do feel free to contact us. So here's my contact info. I'll be more than happy to talk to you or to, provide you with any education or outreach activity if you are interested in doing so. So please feel free for that. And with that, I will take any question.
Thank you very much. Thanks for
Of
sharing with us.
course.
So
Right.
please, a reminder to put your questions in the Q&A. We have a question here. Do lake aerators that spray water into the air increase the risk of aerosol exposure to cyanotoxins?
That's a really, really good question. So if you do have a harmful algal bloom and you have one of those systems that does inject water into the air, potentially you are transporting the impacted water into the air. So that could be. And then the other thing that don't forget, sometimes the mist can be blow over to like sidewalk or shoreline, for example, and the mist could be impacting human as well if you have the mist directly fell onto your body. And that's no different than wading into the water. So again, that would be a concern.
Thank you. I was also wondering, you were talking about sometimes a bloom will survive over winter. Is that because of warmer temperatures or they've just adapted somehow or?
Not necessarily, because they are very strong bacteria. And some of them can survive winter because they are able to become dormant and they can, like, just stay there, just grow slowly. But then some of them actually are very hardy and they can survive in low temperature condition. As a matter of fact, there are some cyanobacteria that are so super good at wintertime. Sometimes that I don't know if anybody had noticed that in the wintertime, you might see like kind of like a pinkish spider web in the ice.
Hmm.
And that is a result of cyanobacteria. So what they do is they use their body heat to melt the ice and create this little tunnel, and they can reproduce and expand themselves into the next area. And so that little tunnel will turn into like a spider web situation. And that is an example of how cyanobacteria are really very, very tolerant, and they could absolutely survive in very hot temperature or very cold temperature. And we have seen multiple incidents that they overwinter and still are going through bloom over winter.
Oh, thank you. We have another question here. Can you tell us more about toxin test strips? Where can we get them?
So the test strip is actually manufactured by the same company that also produced the ELISA testing kit that DEP and EPA recommend people to use, and it is called Golden Golden Standard. And if you type in this word Abraxas, which I can type here for you as well in chat. So Abraxas is like a trademark for the testing. So Abraxas has testing strip that you could actually purchase and you could actually use it in the water testing. However, they're not really cheap. So just want to keep in mind that it's not necessarily very cheap. And you also have a limitation toward the range. So it usually can only do 0 to 5. So if you have concentration over 5, then it wouldn't really show. And then the other thing is its color changes. So it definitely is a little bit tricky.
Thank you. So another
Sure.
question, kind of following up from that is what steps are taken to combat the HABs from the DEP or from the municipalities?
Very, very good question. So at this time, the recommendation is let's start from managing your nutrient. So manage your stormwater, manage your runoff, manage your septic tank, try to reduce your nutrient input. That could be the number one and could be very beneficial and useful to prevent harmful algal bloom from forming. If you do have a ongoing harmful algal bloom, then there are some treatment strategy available. For example, barley straw happen to produce a certain enzyme that could slow down the cyanobacteria from reproducing. So barley straw is actually a more like natural way for treatment. But then there are other chemical that are typically used as algaecide. For example, copper sulfate has been used for generation after generation. For example, peroxide and different kind of peroxide compound has been using as algaecide. So definitely chemical treatment is an option when you have harmful algal bloom. However, I do want to say from my personal experience, it can temporarily kill the cyanobacteria. It could allow you to have your water with low cyanobacteria density, for a while, but it does not guarantee that you will have no cyanobacteria for the rest of the summer or for the rest of the season. From our experience, we do see that it is more like a temporary solution. And then you might have another harmful algal bloom episode a couple weeks or a few weeks afterward. So it is not uncommon for a water body has to do multiple treatment within a same season. And we have heard people do 6, 7, 8 times just within one season. And the accumulation of using the same chemical compound in the water body is absolutely concerning. So I do want to put it out there. If anybody want to do chemical treatment, please do consider that is a possibility. It is not an ultimate solution. It more likely is going to be a temporary solution until the next harmful algal bloom episode. So do have that in mind that could be the case. Because when the cell break, it release nutrient and wait a little bit for them to come back up with a nutrient and just the right condition available, they can, come back up. The other thing to keep in mind is when the cell break open with any type of chemical, it will release the toxin into the water. So you're likely to see even higher level of available toxin in the water. So be very careful about immediately after treatment, how that might be impacting your non-target species, such as waterfowl or fish in the water.
Thank you.
Of course.
And we have one last question, which is like a couple together, but does the surface look different depending on which type of cyanobacteria? And are some more harmful than others? Do they live together, or does one usually dominate a lake or a water body?
Very, very good question. So just like any other plant or any other planktonic group, they also have a preference toward temperature, water velocity, sunlight level, clarity, and stuff like that. So if you look at any single water body, it actually going through the seasonal variation in terms of cyanobacteria and phytoplankton community composition. So in another way to say it is you might see one particular taxa more dominant during the hot summer day, and you might see another cyanobacteria taxa more dominant in the winter, cold winter time, and then maybe somebody else in between. So that shifting could happen based on the condition. And would it be a combination of many? Of course, that would be a possibility. But we have also seen some of the lake with like 100% on single cyanobacteria. So it does happen, so I would say the variation is huge from lake to lake, from season to season. Normally, we'll see that if a lake is dominant with this particular taxa during the hot summer day, very likely we'll have the same one the next year. But, it definitely can vary. Well, going back to the first part, will you be able to see the symptom on the surface of the water body? Not necessarily. It really has a lot to do with your current condition, the wind condition. If you happen to have your wind and current like blow all the cyanobacteria into a corner, or if you have a cove, or you have some sort of irregular shape shoreline, you might have them concentrated at a corner. Or if you have more like a shallow, swampy area, particularly you have like waterfowl, geese like swimming around, you might have a more obvious situation. But a lot of time, they could also be well mixed in your water column. So you might have a high concentration over that 80,000 threshold, but you are not able to visually see, except your water might be slightly more green than usual. But if your water tend to be, yellowish to begin with, then it'll be much more difficult to tell them apart.
Okay. Well, thank you very much for your time and
Of
sharing
course.
expertise. And we will be sharing the recording with everyone who's attended, and also your presentation and the list of links that you shared with us as well.